Welcome back, everybody. So one thing I want to do is just start off with a kind of a review of condensation reactions and hydrolysis reactions. Before we even start that, however, I just want to go over once again the basic form of an amino acid. So you will be, just so you know, you will be asked about this on future quizzes and tests. I'm going to ask you, draw out the fundamental unit of a protein. Well, proteins are made up of amino acids, so you're going to have to draw the basic structure of an amino acid. So if you don't know it yet, now might be a good time to practice. I'm going to go ahead and put it on the board one more time, and then from there we will actually go on with our condensation reaction. So we're going to start off the fundamental unit of a protein, which is an amino acid. So let's start off here. I always write them this way. There are, of course, other ways to write them, but this is just the easiest way for me. So this is the way I do it. Okay. So basic structure of an amino acid. We have number one, we can see right here, we have our amine group, one of our functional groups, right? The amine group. Where we get the name amino acid. The amine group, the functional group, is right here on the molecule. On the other side of that molecule, of course, on the other side of this amino acid, we have over here, we have our other functional group, we have our carboxyl group. Right? Carboxyl group, COOH. And of course, the carbon is double bonded to that oxygen. And then down here, we have our OH. So this whole thing is the carboxyl group. When we talked about carbonyl before, we talked about how there's a carbon double bonded to an oxygen. If it was an aldehyde carbonyl, it was off to the side of the molecule. If it's a ketone carbonyl, it's in the middle of the molecule. So here it looks like almost like a carbonyl. And then down here we have what looks like a hydroxyl group. So carbonyl plus hydroxyl gives us this whole thing called the carboxyl group, COOH. And this part right here, the carboxyl group, is what makes this an acid. This is why this is an amino acid right here. Okay. So all amino acids structured the same way. The only difference, of course, is down here where the radical is. That's why you put the R there. It's kind of like X with algebra. You're trying to solve to find it. Here, depending on what the amino acid is, this will be a different atom or different compound down here on that portion of the molecule. But the whole thing is an amino acid. This whole thing is an amino acid. All right. So as you see, now I've written on the board, I have two amino acids up here. I have this one right here on the left-hand side, and I have this one over here on the right-hand side. And so what we're going to do here is create actually a condensation reaction. So with condensation reactions, we take two monomers, two like 1 unit compounds, and we merge them together to create a polymer. In the process of actually bonding those things together or linking those things together, we do release water in the process, which is why it's called a condensation reaction. So amino acid number one, amino acid number two, they are written identically. All right, so remember, all amino acids have the same structure. It just said this, we just did this, we just talked about this. So each of these two amino acids here, we could say they are completely different amino acids. We could say one is glycine and the other one's leucine. But what we're going to do is, through a condensation reaction, link them together. And when we link lots and lots and lots of amino acids together, we get a protein. So here, this is amino acid number one, amino acid number two. We're going to link them together through a condensation reaction. The condensation reaction is going to release water in the process. So we all know water is formed as H2O, 2 hydrogens, 1 oxygen. And we can actually see that pretty easily right here. On this carboxyl group of amino acid number one, we see an OH. This hydrogen here from the amine group of the next amino acid has another H. So 2 H's, 1 O, H2O, we get water. So I'm just going to draw a little circle around it to show you where the water is coming out of during this reaction. Okay, let's take this little arrow, draw it down. Condensation reaction. We are releasing water in this process. So now that I'm linking them together, now that this OH and this H are gone, they've left, we have to rewrite the newly formed linked peptide. It's going to be called a peptide once they're linked together. So it starts off looking very much the same way. We've got our amine group. That's a hydrogen, sorry. We still got our central carbon here. We still got our radical down here. We still got our outside carbon here. However, the OH is gone now. We still have the O, but the OH is gone. So what I'm going to do is I'm just going to write the O, double bonded right here, above the carbon instead of off at this 45-degree angle. Still the same O, it's just moved up above the carbon. So this is amino acid number 1, just like this one. Same exact thing, except the OH is gone. And now we are drawing the bond to the other amino acid, the one that's over here. So we start off, we've got our nitrogen here from our amine group, but this hydrogen is gone. We still have one hydrogen. So just like with this O, we're just going to place it above the nitrogen. And then everything else just flows as it did before. All right. Now we had, to start with, like I said before, we had 2 monomers. Monomer number 1, amino acid number 1. Monomer number 2, amino acid number 2. Through a condensation reaction that releases water, we link them together and form a polymer. We form a polymer. And this polymer is held together right here in this bond between the original monomer number 1, the original amino acid number 1, and monomer number 2, amino acid number 2. It's now one polymer held together by this bond, and we call that bond a peptide bond. Peptide bond holds those two, well, they were monomers, now it's a polymer. Hold those 2 monomers together to create this polymer. And since we have 2 of these being held together, 2 of these compounds being held together with a peptide bond, we call this whole thing a dipeptide. Dipeptide. Di prefix just meaning 2. Okay. So if we were to continue this on, if this end down here was then bonded to amino acid number 3, in which case it would start looking like this, then we would have a polypeptide. And we just keep building, adding more and more amino acids until we have so many amino acids all linked together that we can actually call it a protein. Usually it's around 52 amino acids linked together. Anything above that, we call that a protein. So once again, this is a condensation reaction. Or you could call it a dehydration synthesis reaction, same term. Condensation reaction, dehydration synthesis reaction. We're getting water out of 2 original monomers, linking them together to create a polymer. All right, so that was condensation reactions or dehydration synthesis reactions, whichever term you want to use. But let's talk about one other type of reaction. Once again, let's talk about hydrolysis reactions. This time, in a hydrolysis reaction, we're actually adding water to something in order to split it. So we're taking a larger polymer, adding water to it, and splitting it into 2 or more monomers. So with this, we could take a polymer such as maltose, which is a sugar, and we can actually add water to it to split it. So if this is maltose here, this whole thing, it's being held together right here in the middle by this oxygen. So both sides of this are being held together by the oxygen. And that bond, that thing holding them together, has a very specific name. It's called a glycosidic linkage. Glycosidic linkage. Holding these two things together, bonding them together. So we have one polymer here. Once we add water to this, it's going to break this linkage and create two separate monomers. So take my blue marker. I say let's add some water to this. Now we're going to get two separate compounds. We had maltose to start with. We add water to it, however. It breaks that glycosidic linkage and gives us two new compounds. And then, let me just do. Nope. Hold on. There we go. All right. So we had just oxygen here. We end up adding H2O. So there's our two H's, H2. We already had one O. There's the other O. So we create two separate compounds, two monomers. They are no longer bonded together. They are no longer linked together. As one polymer, this was maltose. We added water to it. We split these apart. Now as two monomers, they are glucose. They are both glucose. Both glucose. So this is a hydrolysis reaction. One polymer, water added to it, breaks apart the glycosidic linkage, creating two separate monomers. Hydrolysis reaction. Hydro meaning water, lysis meaning split or break. We added water to this to split or break that polymer into two separate monomers.